Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel Diversity of Plants Zone. Before starting my today's video, I request you all to please subscribe my YouTube channel. Today's topic of my video is about taxonomic characters. So let's start the video. First of all, I will describe about introduction. All the biological organisms have earned distinct characters by themselves. Characters contributing to a taxonomic description is known as the taxonomic character. Taxonomic character is any physical structure or behavioral system that can have more than one form. Character state, which potentially provides phylogenetic information. Taxonomic character is defined as any attribute of a member of a taxon by which it differs from a member of a different taxon. A characteristic by which members of two taxa agree but differ from members of the third taxon is a taxonomic character. In more general terms, character is any attribute or feature of an organism or a specimen that can be measured, assessed or weighed. Next is character state. Character state, it is the various patterns of their expression. An expression of the character is called character state. Characters and character state. For example, character is leaf arrangement and character states, for example, alternate, opposite or rolled. Next are molecular characters. Molecular characters, for example, nucleotide bases. Example is adenosine, cytosine, thymine and guanine. Second is amino acid codons like ACC, CGT, GAT, etc. Characters can be qualitative and quantitative. First of all, I will describe about quantitative characters. Quantitative characters are those characters that can be assessed by size, length, weight, etc. Next is qualitative characters. Qualitative characters are those characters that are relating to form that can be expressed. Qualitative characters are such characters as leaf arrangement, for example, opposite versus alternate leaves, placentation types like exile and praetor. Often the distinction between quantitative and qualitative character is more apparent than real. Many qualitative characters may also be expressed quantitatively, for example, leaf shape in width and length ratio. Application of the quantitative and qualitative characters. Quantitative characters appear to be more frequently used in the lower taxonomic categories at specific and intraspecific levels. Thus, species may be separated by the leaf size, corolla length, etc. Qualitative characters are often used at higher levels in the hierarchy, for example, family. Good and bad characters. All characters are theoretically of use in classification, but experience has shown that their value or use varies according to the type of taxonomic activity which is envisaged. Good characters are those that are not subject to wide variation within the sample being considered, do not have a high intrinsic genetic variability, are not susceptible to environmental modifications, and show consistencies, for example, agree with the correlation of characters existing in a natural classification that was constructed without their use. Next is diagnostic characters. Is a shortened description covering only those characters, diagnostic characters, which are necessary to distinguish a taxon from other related taxa. Taxonomic characters basically are classified into two types. First is continuous and the second is discontinuous characters. First of all, I will describe about continuous characters. When the variations exhibited by a taxon displays a continuous gradation without obvious break, we call it continuous variation. Continuous characters are related to those variations between species due to genetic and heritable changes. These variations result from the influence of many genes and metabolic interactions. In continuous variation, there is a complete range of the measurements from one extreme to the other. Height is an example of continuous variations. Individuals can have a complete range of heights, for example, 1.5, 1.51, 1.52, 1.525, etc. meters high. Weight, body size, hand span, shoe size, and milk yield could be other examples of the continuous variation. Second is discontinuous variation. Discontinuous variations are different from continuous ones in that the latter variation is always caused by the genetic variation. In the former, it can be purely environmental in origin, while the consequence of the conditions encountered during the development. When the variations exhibited by a taxon display obvious break or discontinuities, we call it discontinuous variation. It is through these gaps the delimiting lines between taxa are drawn. For example, shell coloration in the land snail. In taxonomic practice, it is extent of gaps that usually determines the taxonomic ranks. The greater the break or the gap, the higher is the taxonomic rank attributed. About 3 million of the species of organisms that have been discovered so far are on the basis of such discontinuity and would remain so unless and until further investigations reveal intermediates. Next topic is taxonomic information, which is also called taxonomic evidence. Taxonomic characters can be drawn from any part and phase of development of organism. In practice, overwhelming reliance is placed on morphological characters. However, the following are the major sources of the taxonomic information or evidence. First is morphological and anatomical evidence, embryological evidence, cytological or chromosomal evidence, phytochemical or chemical evidence, information from breeding systems, and geographical and ecological evidence. So first I am going to discuss about the morphological and anatomical evidences. Taxonomists still rely today to a very large extent on the morphological and anatomical characters. The reasons why the morphological characters are more often used than the other characters are: first reason is morphological features have the great advantage that we can see them easily and therefore appreciate their variability with much less facility than with other kinds of features. Even on herbarium species with which most taxonomists' work has to be performed, morphological characters can be used easily. Second is embryological evidence, the early development of organisms. Some of the embryological characters that are considered to be taxonomically significant include: first is anther. Anther taxonomic characters of the anther include number and arrangement of the thecae, type of the wall development and organization of the pollen grains. Second is ovule, development and structure of the ovule, which include number and structure of the integuments, presence and absence of an aril. Third is cytological or chromosomal evidence. Taxonomically important chromosome data are chromosome number, chromosome size and shape, chromosome structure, chromosome behavior in meiosis, and DNA content. Variations in chromosome number have been characteristics of the herbaceous dicots, and woody dicots show much less diversity. Chromosome numbers in flowering plants vary from n is equal to 2 in a species of the Asteraceae to n about 250 in the Kalanchoe. Next is phytochemical or chemical evidence. Two types. First is micromolecules. Micromolecules have low molecular weight compounds and are products of plants' secondary metabolites. Include flavonoids, terpenoids, alkaloids, betalains, and glycosinolates. Next is macromolecules. Proteins and nucleic acids are included in it. They are found in nucleus, mitochondria, chloroplast. These are high molecular weight compound, and these are the part of basic metabolic machinery of the plant cells. Fifth is information from the breeding systems. The breeding systems of the plant is the mode, pattern and extent to which it interbreeds with the other plants of the same or different taxa. Breeding system determine if the pollen grain can germinate on a receptive stigma, penetrate the style and effect the fertilization. Inbreeding, the mating of individuals or organisms that are closely related through common ancestry as opposed to outbreeding, which is the mating of unrelated organisms. Next I am going to discuss about breeding system. It is taxonomically important for three reasons. The reasons are the extent of interbreeding largely defines the pattern of variation and hence the delimitation of taxa. A knowledge of the breeding system frequently helps to understand taxonomic complexity, although it does not solve the problems associated with it, and a study of the breeding system is often vital in unraveling evolutionary relationships. Next I am going to discuss about the hybrid. Formation is common in plants and there are about 70,000 species of hybrid origin, most between 2 or among 3 or more species belonging to the same genus. And there are also the hybrids between species from different genera but not from different families. So how to recognize the hybrids? This is the question. A hybrid is usually intermediate in appearance between its parents. Whenever reproductive isolation develops, speciation has occurred. Speciation is the evolutionary process by which the new biological species arise. What are the isolating mechanisms? There are several factors that operate in the wild which prevent hybridization. They are, first is pre-zygotic mechanisms. So first I am going to discuss about the geographical isolation. Two species are separated geographically. This is called geographical isolation. Second is ecological isolation. Two species occur in the same general habitat but are separated ecologically, occupying different habitats. This is called ecological isolation. Third is seasonal isolation. Two species occur in the same locality but flower at different seasons. This is called seasonal isolation. Fourth is temporal isolation. Individuals of the different species do not mate because they are active at different times of the day or in the different seasons. This is called temporal isolation. Fifth is ethological isolation. Related interfertile species are pollinated by different sorts of vectors or any particular forage. An individual pollen vector keeps to one species only. This is called ethological isolation. Next is mechanical isolation. Related interfertile species have differently structured flowers, which make it difficult for pollen vectors to transfer pollen to the stigma of flowers other than those of the same species from which it was obtained. It is called mechanical isolation. Next is breeding behavioral isolation. It occurs when populations of the same species begin to develop different behaviors that are not recognized or preferred by members in another population. Potential mates meet but choose members of their own species. Next is gametophytic isolation. Cross-pollination takes place but pollen tube fails to germinate or to reach and penetrate embryo sac of the female parent. This is called gametophytic isolation. Next is gametic isolation. The pollen tube releases male gamete into the embryo sac but gametic fusion and or endospermic fusion does not take place. This is called gametic isolation. Next is post-zygotic mechanisms. First is hybrid incompatibility. The zygote or immature embryo stops development so that a mature seed is not formed. So this is called hybrid incompatibility. Next is hybrid inviability. Seed germination occurs but the resultant F1 hybrid generation dies sometime before its production of flower, and this is called hybrid inviability. Next is non-fitness of the hybrids. The F1 hybrids reach maturity but are not successful competitors in the wild. No suitable ecological niches formed. So next is F2 hybrid sterility. The F1 hybrids are fully viable but sterile and doesn't contribute to future generations. So next is F2 hybrid inviability or sterility. The disability is delayed until the F2 generation or even later. Next is non-fitness of F2 hybrid. Disability delayed until a later generation. Alternatively, isolating mechanisms like 1 to 7 may be described as external and 8 to 15 as internal. Next is geographical or ecological evidence. Ecological evidence. Ecological data are different from other types of comparative data in taxonomy. They deal with plant environment interactions. The interactions are the net effect of all features of plant with all aspects of their environment. There are two basic types. First is abiotic. Abiotic, as we know, physical rather than biological, not derived from living organisms, and it is devoid of life, sterile, such as the soil. So next is biotic. Biotic describes a living component of ecosystem. For example, organisms such as the plants and animals. Other examples include water, light, wind, soil, humidity, minerals, gases, all the living things like autotrophs and heterotrophs, which include plants, animals, fungi, bacteria. Biotic component or biotic part of the environment include aspects such as herbivores and competitors. So one of the most commonly used ecological data in taxonomy is distribution, which is also regarded as geographical evidence. The associated plants in the local environment are very variable as comparative data, which are usually given as general plant communities described by dominant taxa. Next is pattern of the geographical distribution. There are 4 patterns of the geographical distribution. I will discuss it one by one. First is allopatric. Taxa which occupy mutually exclusive geographical areas. The isolated population undergo genotypic or phenotypic variations or divergence. Second is peripatric. In this, new species are formed in isolated small peripheral populations which are prevented from exchanging genes with the main population. The isolated individuals quickly become incompatible with their former species and have become new species. Next is parapatric. In this zone of the two diverging populations are separated, but there is only partial separations like the edge of the forest or the snow line on the mountain. Last is sympatric. Sympatric occurs in population that lives in the same geographical area, less common than allopatric speciation. Happens when gene flow is diminished by polyploidy, habitat differentiation and sexual selection. Next topic is disjunction and vicariance. The most taxa are found fairly continuously throughout their area of distribution, but some have distribution patterns that are interrupted by considerable areas from which the taxon is absent. The interrupted distribution pattern is called disjunction or disjunct distribution. Disjunction may originate in 2 or 3 ways. Long distance dispersal of the taxon from one area to another. Second is disjunctions may represent the former wide continuous distribution patterns having been depopulated, sometimes because they were covered by sea. Another possibility that the taxon may arise independently in its separate areas of distribution, thus exhibiting evolutionary parallelism or convergence. Next is Vicariance, VICARIISM, VICARIADS or VICARIANTS. Two similar taxa occupying separate geographical or ecological areas are known as vicariants or vicariates. The phenomena by which the two similar taxa occupy separate geographical or ecological areas is called vicariance or VICARIISM. The difference between the vicariance and disjunction is one of a degree. Next topic is identification. Identification of an unknown organism is the determination of it as being similar to or identical with an already known element. It is the naming of an organism or part of an organism by reference to an already existent classification. There are four methods of identification. These are: number 1, expert determination; second is determination by the recognition; third is determination by comparison to preserved and well authenticated specimens; and drawings, illustrations. In terms of reliability and accuracy, the best method of identification is first one, which is expert determination. In general, the expert will have prepared treatments like the monographs, revisions and synopses of the group in question. Recognition approaches expert determination in reliability. This is based on extensive past experience of identifier with the plant group in question. The third method is by comparison of the unknown with named specimens, photographs, illustrations or descriptions. Even though this is reliable method, and it may be very time consuming or virtually impossible due to the lack of suitable materials for comparison. The use of keys or similar devices like synopses, outlines, etc. is by far the most widely used method and does not require the time, materials or experience. Next topic is about the keys which are used for identification. So keys are of two types. First is single access or sequential keys. Have a single starting point or fixed sequence. These are called single access or sequential keys. So what is meant by the sequential keys? These are nearly always written in the form of dichotomous keys, so familiar in the botanical and zoological works. Next is multi-access keys. They can be commenced at any position. Multi-access keys are usually produced not on pages in a book, but on separate punched cards. Single access or sequential keys. Sequence keys are nearly always written in the form of dichotomous keys. Now the question is, what is meant by the dichotomous keys? Dichotomous keys are the devices consisting of a series of contrasting or contradictory statements or propositions in requiring the identifier to make the comparisons and decisions based on the statements in the key as related to the material to be identified. The dichotomous keys consist of a series of the couplets or mutually exclusive pairs of statements. Each statement or lead of a pair leading on to a further couplet. The dichotomous or sequential written in one of the following ways. First is bracketed key and second is indented key. The essential difference between these two layouts is that in the indented key, all the possibilities arising from the first lead are dealt with before the second lead is mentioned. Bracketed key are known as parallel keys and keep the two leads of every couplet together and easier comparisons of the alternatives. So, the bracketed keys utilize the space in each page efficiently and so they are cheaper to print. However, the indented key has the advantages when the key is short as the pattern of character is clearer. But when the key is long, there is much wastage of page space and the user has to turn pages to find two halves of the couplet. There is wastage of paper because the keys utilize the right side of the paper and so they are expensive to print. Next topic is suggestions for construction of keys. First is identify all the groups to be included in a key. Prepare a description for each taxon. Select key characters with contrasting character states. Use macroscopic morphological characters and constant character states when possible. Avoid characteristics that can only be seen in the field or on specially prepared specimens. For example, use those characteristics that are generally available to the user. Prepare a comparison chart. Construct strictly dichotomous keys. Use parallel construction and comparative terminology in each lead of a couplet. Use at least 2 characters per lead when possible. Follow key format indented or bracketed. Start both leads of a couplet with the same word, if at all possible, and successive leads with the different words. Mention the name of the plant part before descriptive phrase. For example, leaves alternate but not alternate leaves. Place those taxa with numerous variable character states in a key several times when necessary, and construct separate keys for dioecious plants for flowering and fruiting materials. Select appropriate keys for the materials to be identified. The keys may be in the flora, manual, guide, handbook, monograph, or Vivian. If the locality of an unknown plant is known, select a flora guide or manual treating the plants of the geographical area of that geographical area. If the family or genus is recognized, one may choose to use a monograph or Vivian. If the locality is unknown, select a general work. Read the introductory comments on the format details, abbreviations, etc. before using the key. Read the both leads of a couplet before making a choice. Even though the first lead may seem to describe the unknown material, the second lead may be even more appropriate. Measure several similar structures when measurements are used in a key. For example, measure several leaves, not a single leaf. Try both choices when dichotomies are not clear or when information is insufficient, and make a decision as to which of the 2 answers best fits the description. And verify your results by reading a description, comparing specimens or comparing the specimens with an illustration or authenticity. So, if you like my video, please like and subscribe to my channel. Thanks for watching. Thank you so much for your time and appreciation.